So I am a pharmacologist by training.
Okay.
I attended a HBCU in South Carolina, Claflin University.
Yep. I know
For my
Claflin well.
undergraduate, I studied biochemistry. I'm originally from Nigeria, so Claflin brought me here to the States back in 2007 on a full scholarship from the Honors College, without which I really would have not been able to afford college in the US.
Yeah.
Coming here, I thought I wanted to be a medical doctor because growing up in Africa, like when she likes science, everyone thinks you need to be a doctor and engineer.
Right.
But I knew that I was fascinated by biology, but I also knew that I didn't necessarily want to treat people in a hospital setting. So it was about like, what is that in between where my scientific curiosity can be channeled into what I now know to be drug discovery. And so it took me a minute to actually put a label to what I wanted to become. As a pharmacologist, it wasn't something that people really understood. And so going through Claflin, I came in as a first year biochemistry major in the Honors College. In the second year, I thought I wanted to declare pre-med. I talked to a couple of people that thought perhaps I should go to pharmacy school. So I kind of left the pre-med track, but I remained as a core biochemistry major, giving me the optionality to pursue any number of tracks. Internships were instrumental in helping me figure out what the path should be for me. The summer after my freshman year, I had the opportunity to do my first summer research experience at the George Washington University through the Howard Hughes Medical Institute. And I really, really enjoyed that opportunity because it was my first time doing research. I had never done research back home in Nigeria. I had not done that type of research at Claflin, just given that I had only completed my freshman year. And it was genetics research, studying how genes and mutations in genes contribute to different types of phenotypes in a type of fruit fly called Drosophila melanogaster. So it was exciting. I got to do my first PCR. I got to hold a pipette for the first time. I got to do DNA prep, and that experience was instrumental in a lot of ways. So coming back on campus to my sophomore year, I knew two things about my research experience that freshman year summer. First, I knew that I enjoyed it, but I also knew that I wanted to do something that was more translational. I kept yearning for an opportunity to work on something that wasn't necessarily about fruit flies, but was something that had to do with like a disease state that I was passionate about treating. And so the summer after my sophomore year, Claflin started a partnership with Yale University via the Sackler Institute. So Richard Sackler and his family had put together a foundation, a fund to help HBCU students attain research experience at some of the top universities in the country. So via that program, I got to spend the summer after my sophomore year at Yale, working with Andrew Nwankwo over in the biology department, studying a disease called, I think it was copper-induced amyloidosis, this idea that when patients have kidney disease, oh, by the way, kidney disease is most prevalent amongst African Americans and people of African descent. But when people have kidney disease, they frequently go in for a treatment course called dialysis. And the hypothesis that we were studying was that patients could actually develop a buildup of plaques in their kidney, a type of amyloidosis that was induced by the metal that they encounter on the surface of the dialysis machine. So we're kind of studying that phenomenon in an in vitro setting using different metal ions like copper. And so that was yet another formative experience from the perspective of, okay, not quite working on a drug project just yet, but one step closer towards a translational biology experience. So coming back to Claflin for my junior year, when I finished my junior year summer, I really targeted pharmaceutical companies. I was like, okay, I finished 2 years of core biochemistry courses. I've done 2 internships, summer research experiences at 2 great institutions. I really want to break into the pharmaceutical industry. And obviously it was difficult, right? Being in South Carolina, there are really not many pharmaceutical companies in that area. And frankly, not many alums of Claflin had gone the pharmaceutical path. So there weren't a lot of people to talk to. And I just was just putting applications out there into the ether. Not really sure if anybody was reading it. I'd literally go on Google and be like top pharmaceutical companies, and Google will be like, oh, number one, number two, number three, and I'll just start applying to each of them. I was fortunate to end up with 2 internship offers that summer. The first one at Eli Lilly out in the Indianapolis headquarters, and then the second one at Novartis here in Cambridge, Massachusetts. I ended up deciding that I wanted to spend more of my time doing cancer research, but the opportunity at Eli Lilly was fantastic. And so I actually did 2 internships in the same summer. I spent about 6 weeks working in the biotherapeutics discovery research group at Eli Lilly, where that group was tasked with optimizing a drug that is now known as Trulicity for type 2 diabetes. And we worked on things like, helping to make protein modifications, because it's an antibody based drug. And so helping to make modifications that will help the drug sort of have a longer half-life. So you don't have to take so many courses of the drug if you're diabetic. And so it was really fascinating working on that project, albeit for a short period of time. But I came to Cambridge, Massachusetts for the first time in the summer of 2010. I spent about 12 weeks at the Novartis Institute for Biomedical Research. And that experience really opened my eyes. It was a challenging experience. I was in the oncology cell biology group, and we're focused on basically understanding the mechanistic basis for drug candidates that Novartis was moving forward. So we had drugs that they were like, oh, this drug shrinks tumor size in mice, or it prevents metastasis. But what is the underlying signal transduction pathway? And it was fascinating teasing apart, proteases and kinases and a variety of cellular factors that might be playing a role in terms of contributing to the downstream phenotypic effect that you were seeing, whether making cancer cells shrink or whatever that might be. So I really love that experience. And I think it was there that I actually decided I was going to do a PhD instead of an MD. Because for the first time I was like, okay, these guys speak my language. So what I really want to do is to make drugs. And I want to do it from a rational drug design perspective, which means that I want to really understand how the drug is having the molecular effect, the molecular pathways that underpin the drug action. I want to really understand that at a really deep level. And so while I was finishing up my internship that junior year here at Novartis, I took the time to start visiting schools in the area. I visited Harvard, I visited Cornell, Columbia, and then I went back to school to finish out my senior year at Claflin. In parallel, I started applying to PhD programs, to the GRE that fall of senior year, to the GREs, completed all my PhD applications, put them in, did interviews in February of the next year, got into Tufts University. I like Tufts program in pharmacology and experimental therapeutics because it's a program that really focuses on, training you to understand, just the deep biology behind drug mechanisms, but also focused on people who wanted to go into industry. I knew from jump, and even till this moment, that I was not cut out for academia. I've never wanted to be.
How did you, if you don't mind me asking, how did you know that though? That's a very strong statement. And I think it's a lot of graduate students, perhaps even postdocs that might be listening to you, might, want to know, well, how did you figure that out? What was it that told you that?
I think that right from when I was a kid, I'd always looked at my career from a practical lens. And my mother tells a story, at least she told the story to me one time. When I was 7 or 8 or something, I was in Nigeria and I had malaria and I had typhoid at the same time. And I was taking a cocktail of like what must have been 8 to 10 pills at a time. And I was so skinny and so emaciated and so sick from having these two parasitic illnesses at the same time. And the medicines were just so many. And what I started doing at some point, because I got tired of trying to swallow 8 pills, the ones that were capsules, I would open them and like dump out their content. And the ones that were like tablets, I would crush them up. Basically, I was trying to condense 8 medicines or 8 pills or whatever into one thing. And then I reassembled them into one capsule, and I wanted to take just that one. So you can say that I have been doing drugs for a minute. Put it that way. But I think that how I knew that I wanted to discover drugs. I was fascinated, and I still am fascinated anytime I see a molecule. Like I would just hold it up. Like it could be anything. It could be a Tylenol. It could be anything at all. I've always been fascinated about that. And I think that's why some of my advisors at Claflin thought I should go the pharmacy route. I had a lot of academic advisors who said, "Oh, Jen should go to pharmacy school." I got that advice a lot. So I think my sort of natural gravitation towards just being fascinated by molecules and drugs and tablets and capsules, I knew I was going to end up doing something with pills. And I just didn't see how that would happen in academic context. And the time I went on to spend in pharmaceutical companies, even as a junior or college junior, and then again as a college senior, because I returned to Novartis for another year, solidified in my mind that, the way of thinking, the pragmatic way of thinking about science in industry was more me than the very sort of puristic way that people in academia think about it. And I think academic research is important. Without academic research, there would be no industry research. It's not to say it's one or the other, because fundamentally everyone who's in industry came from academia, right? We went and got our PhDs and so on. But I found that there was something about the speed with which industry moves, the very practical, data-driven nature of then at Novartis, it was all about the data, go, no go decisions and make purely based on data. But in academia, you'd see projects just kind of like string on and go on and on and on just because the NIH is funding it. And people studying things just because the NIH is funding it kind of wasn't my beat. I wanted to work on something because I could actually see a path forward for it. Clinic. So I think I'd always known that I would end up in a corporate setting, even from when I was a young child.
So, wow, that's fascinating. Let's go back a little bit to your undergraduate experience at Claflin. And by the way, Claflin University is a UNCF member institution. We're very proud of that, and certainly has a tradition of producing brilliant scientists and STEM professionals like yourself. But talk to us about your Claflin experience, and how do you think that experience prepared you for where you are today?
Oh man, I don't know if there are enough words to say thank you to Claflin University. I don't think really there are enough words. First of all, very practically, I went to school for free. Okay, so let's begin there. I had 4 years of a merit-based scholarship from the Honors College. And so how did it prepare me? As a young kid, I was 17 years old when I started college. I came from Nigeria. I didn't have family here. And Claflin provided a soft landing for me, being an HBCU, coming from a country where pretty much everybody is Black, and then going to a school where pretty much everybody is Black. It allowed me a softer landing into the United States. So there's that piece. However, when I started going out to do scientific research, whether it was at George Washington or at Yale, I was like, oh, one of these things is not like the other. Okay. I think I didn't have that appreciation for like race relations coming here, because you never think about the fact that you're African American. You never think about the fact that you're Black, because it's like when you're in a place where everybody's Black, it's like, okay. So Claflin offered me first an opportunity to get a great education. I wanted to be a scientist. It gave me the landing pad to do that. It gave me the financial resources to do that. I didn't have to worry. I just had to keep my grades really high. And it gave me a network of teachers who pushed me really hard. And even when I didn't have the right label for what it is I wanted to become, even though it took me a minute to figure out what my own path was, because it wasn't medical school, it wasn't pharmacy school. Even at some point, somebody recommended a nursing assistant program, a physician assistant program. It wasn't all of those things. While the answer wasn't made immediately clear to me on the very first day of freshman orientation that I should study pharmacology as a PhD, the tools were there.
Yeah.
So what Claflin does really well, I was in the School of Natural Science and Mathematics for 4 years. The dean of the School of Natural Science and Mathematics, Dr. Verley Teasdale, she was someone that I could go and talk to and just be like, here's what I'm thinking. Here's how I'm thinking about this. Here are all these crazy ideas I have. So definitely shout out to Verley. Also Angela Peters, who is the chair of the Department of Chemistry, where I was a student majoring in biochemistry. I worked with her very closely. First, she taught me a few classes, but also being part of the American Chemical Society, which I ended up becoming president of my last 2 years at Claflin. Through that network of ACS, we got to be exposed to a variety of career opportunities. We got to see a lot of companies. Pretty much every single pharmaceutical company is a member of ACS, right? So ACS was a platform through which I got to interface with a lot of pharmaceutical companies whom I either ended up working with or collaborating with in some capacity. And the curriculum, the curriculum at Claflin emphasized both, obviously, the theoretical aspect of things, but also getting you to think about research. And while we didn't have, the type of labs we have at Tufts Medical, where I did my PhD, the labs that we did have gave us that foundation to begin thinking about what the next steps would be. And in my junior year, I got to work with my academic advisor, Nicholas Panasic, who is a professor in biochemistry, and he had just come from the University of Wisconsin in Madison. And I would say, from a depth of biochemistry perspective, nobody pushed me harder than Nicholas Panasic. He had the opportunity many years later to come up to Boston to attend my PhD defense about 7 years after I left Claflin or so, left his class as a sophomore. And so coming up and seeing me at my PhD defense at Tufts Medical, he said to me, oh my goodness, did you imagine this day, thinking back to when you were a junior, taking my 4-page biochemistry exam? I was like, yeah, those exams of yours were something else, but yeah, sure. I really appreciate the preparation. So yes, I think from an academic training perspective, the experiential learning in terms of research preparation and the tools to figure out what your path would be, and obviously the financial resources, those are just some of the many, many ways that Claflin is responsible for where I am today and hopefully where I'm going from here out.
That's wonderful. Thank you. And I appreciate the passion which you speak about your alma mater. Talk to us about your graduate experience and getting your PhD and transitioning from PhD to postdoc. What was that experience like? You were at Tufts in Boston. I've been in Boston. The winters are cold. The wind is blowing. But talk to us about that experience and also from the perspective of being a woman. How has your experience as a brilliant African woman in America going through graduate school, one of the top flight programs, what were some of the things that you had to deal with, and how did you deal with them?
Yeah, I think first of all, Tufts in my first year was very challenging. And the reason it was challenging was because so I studied biochemistry in undergrad at Claflin, but then I signed up to do a PhD program in pharmacology. Those two things are not quite the same thing. And frankly, pharmacology is not a discipline that is taught at the undergraduate level. Right. So a lot of the principles of pharmacology, metabolism, pharmacokinetics, pharmacodynamics, I was hearing it for the first time. If I would have gone to a PhD program in biochemistry, it would have been probably easier landing because I did a bachelor's in biochemistry. I had seen some of those concepts.
Speak the same language. Yeah.
Right. I think that it didn't even have to be Tufts. Wherever I went, as long as you're doing this sort of multidisciplinary thing where you trained in one area and now you're trying to train in a different area, you should just prepare yourself for the possibility that you need to put in some extra hours. And that was certainly the case for me. And one of the things when I got to Tufts Medical School in the fall of 2011 to begin my PhD, my class of PhD students in the pharmacology program, there were only 4 of us. And the other 3 people, one was a pharmacist and she had a PharmD and she had practiced for 10 years and she had memorized all the drugs in the world and she knew everything. And the other person had spent like, I think about 10 years working at Sanofi. And then the third person in the class, basically, I was the youngest in the class, first of all. I think I was the youngest to ever come into the program, and certainly the youngest to ever graduate from that program, and the only Black person to have graduated from that program. And so there was a thing there where many of them, because especially by having a pharmacy degree, a PharmD, you are quite knowledgeable about aspects of pharmaceutics, drug formulation, and all of these things we had to learn in our first year didactic courses. So first year was hard. There's just no way to put it. It was hard. I was coming directly out of an undergraduate program. Everyone else had some advanced degree. And I think that's one thing I'd recommend, or I'd advise those who are listening. If you're going to do like a, I wouldn't call it a career path change, but if you're going to go into a slightly different scientific discipline like I did, you might consider taking like a gap year. I don't regret going directly. Like I graduated in May, I moved to Boston like the middle of May. I did another internship at Novartis that summer, and then I started my PhD like 3 months later. I don't regret that, but if I had to do it again, might I have taken a year or two and done maybe a post-bac program, or done any number of things just to frankly, just get my bearings a bit, as opposed to coming out of a bachelor's program and going directly into a PhD program, one in pharmacology that I didn't have prior training in. So it was challenging from that perspective. I felt like, I'll come out of class and feel like I needed to put in another 2 hours just to go over the material, just to really internalize it. But after the first year, the way that the PhD program at the medical school, I emphasize the medical school because the Graduate School of Arts and Sciences at Tufts is slightly different. So I emphasize the medical school because that's where I got my PhD. At the medical school, most PhD programs are 5 years long. The program in pharmacology, for example, starts out with a year of didactic classes where you're mostly in class learning, concepts across a variety of biomedical sciences. And then you also simultaneously are spending time rotating through a couple of labs. So I did 4 rotations in my first year. And then in the second year, committed to one lab, and would then go on to do my doctoral thesis in that lab, and probably took 1 or 2 additional courses, elective courses, in my second and third year, et cetera. So the first year was challenging. The first semester was especially hard, but the semester after that, the next one, I felt like it started to make sense. By the time I got to my second year and I was done with classes and I could focus exclusively on research, it got even better. So from the perspective of the academics, that was what made it difficult for me. In terms of the adjustment, Boston was also different, right? Even though I had been at Boston the previous year, interning at Novartis, I had seen what the environment was like. Boston is just different, it's diverse yet homogenous. It depends on who you ask, and it depends on what you're thinking about. So that's how I like to describe Boston. It's diverse yet homogenous, and you're just gonna have to find your crew when you come here. From the perspective of the thing that I think was most visible to me about being in Boston, being at Tufts, was the fact that I'm Black, right? You just know it. Like you just know it. It's present in your mind because you would not see anybody else who looks like you. You could go an entire week, and you have to be comfortable with that. Again, that took a bit of an adjustment. Coming from Nigeria, going to a HBCU, and now for the first time coming into this environment where I am now officially a minority. I didn't feel like a minority at Claflin. I didn't feel like a minority where I was born, and so on. So I think from the perspective of being from the perspective of being a woman, not so much. I didn't actually. The PhD program at Tufts Medical School enrolls more women, graduates fewer women, but enrolls more women to begin with. So there's attrition along the way. So I didn't particularly feel different, but I think the thing that made me stand out the most in my own mind was just the color of my skin.
And when you got to your postdoc phase,
I didn't do a postdoc, and we can talk about why.
Yeah, well, that's what I was going to say. And you had to make some decisions to which direction to go in.
Yeah.
Walk us through how you were thinking and what told you to go one direction versus another.
Right. I think that this is probably where I should introduce to people the fact that I'm an entrepreneur and a business person. Because this whole time you're hearing about me, you're like, oh, okay, she's probably someone who wears a lab coat. I haven't worn a lab coat in a minute. So I talked about how I went through a difficult first year, settled in a lab in my second year, was fortunate to be awarded the Howard Hughes Medical Institute National Fellowship, which meant I didn't have to worry about my PI needing to pay for my stipend. Howard Hughes was paying that for me. I would recommend to any student, if you could bring in your own funding as a PhD student, do it. It gives you a lot of latitude to pursue any number of areas. I ended up doing the definitive research for my thesis at the Boston Children's Hospital at Harvard Medical School, actually not at Tufts. And a lot of that was because I had my own funding from Howard Hughes. So I would definitely say, if you're a student, whether it's an NIH, F31, or Howard Hughes, or any number of prestigious fellowships out there that fund you for 3 or 4 years, where you can take the fellowship to any lab you want. You're not tethered to one person. Not like I bounced around. I didn't bounce around. I stayed with the same PI the whole time. But having that flexibility was very helpful. So that's just a plug-in. Apply for funding because there are lots of them. So
what did the Howard Hughes Award give you? Was a stipend and research funding?
Yeah, it was about $140,000 for 3 years. It paid my annual research stipend, which at the time at Tufts, I believe was $31,000 for a PhD student. It also paid for all of my, whatever Tufts was charging for curriculum training for PhD students. It also provided some reagent research money to my PI, which he was very happy about. It's actually paid for everything for me as a PhD student over a 3-year period. That particular fellowship you become eligible for in your second year or third year. And it funds you for up to 3 years. And as part of that, you also get to come to Howard Hughes Medical Institute out of Maryland to present your research. So there are lots of fellowships like that. There's the F31 with the NIH. I didn't apply to that one. You cannot hold 2 of these because it's such large fellowships. So once I got the Howard Hughes in my second year, I was the first Tufts student to get this particular fellowship. Once I got that, I didn't need to apply for additional funding. I also know that UNCF has one. I think it's the UNCF Merck or something like that.
Yeah, that used to be. And now we're with the Ernest E. Just Life Science, Postgraduate Life Science Fellowship that's being sponsored by Bristol Myers Squibb.
Okay.
So, yeah, we do have fellowships that are available. So I'm glad you put in a plug for Howard Hughes. Definitely, that's very attractive as an award for graduate students and certainly postdocs as well.
Right. Yeah, no, totally. Apply to any fellowship. They really help you. They stay on your CV. They help you with jobs. They help you with landing the next fellowship, the next postdoc, whether you're going academia, an academic path. They just stay on your CV, like one of the most notable aspects of your doctoral training. So, so I'm glad that I was able to apply for that, and I'm thankful that I was awarded that fellowship in my second year. So that fellowship covered the rest of my training, years 3, 4, and 5. I think by the end of year 5, because I finished, I think at 4 years and 6 months or something like that, I actually returned 6 months of funding back to Howard Hughes because I didn't spend exactly 5 calendar years. So some of that funding was returned to Howard Hughes. So speaking about, you asked a question, how did I decide what to do next? So part of what gave me flexibility. So now I'm a 3rd-year student. I have my own funding the rest of the way. It allowed me to just focus on doing my research and thinking about what was next. And one thing I started to realize, actually this dated back to, so I talked about how when I graduated from Claflin, I moved up to Boston, but the summer between graduating from Claflin and starting my PhD at Tufts, I did a second internship at Novartis, this time in a group called Chemical and Pharmaceutical Profiling, which helps sort of all the therapeutic areas at Novartis think about how to formulate your drugs, how to get your drugs in like a stable state, and how to think about essentially that transition from the research to the development phase. That group had interfaced quite a bit with another group, Alliance Management and Business Development. So I started to see this breed of people who, they just look cool, by the way. Like, they were scientists, but they were very articulate and very eloquent. And in talking about the business implication of science. So I started hanging around some of them, and I realized that there are actually scientists who are not at the bench. And I was like, oh, is that possible? You could be a scientist, but not at the bench. I was like, oh, okay, let me hang out with this group a bit more and learn a bit more. And so, long story short, in spending time with the business development people who are focused on bringing external innovation into NIBR, a new technology, a new platform, a new molecule, basically trying to show up Novartis's pipeline by bringing in external technologies, I was very fascinated. And I actually played a role in evaluating a certain technology that we were looking at from one of the top academic institutions out on the West Coast. I did the wet lab experiments to test that technology and see whether Novartis should in-license that technology. So when I finished that internship, I walked away from NIBR thinking, there might be another path. There might be another path, but I wasn't sure what it was. But I was like, let me go get my PhD real quick. So I went and I began my PhD program. I sort of forgot about that thought for a minute, because first year, like I said, of PhD was so hard. But by the time I got to second year, certainly third year, I had reached what I call cruising altitude, because I liken a PhD to taking off on a plane. The takeoff is very rough. That's like your first year and a half, maybe even the second year. But at some point you get to cruising altitude, the pilot is like, you can take off your seatbelt, you can walk around. When I got to that phase where I felt like my PhD research was on like autopilot, I started to think about what to do next. And I revisited some of those conversations that I had with people in the pharmaceutical business development side. And a lot of them advised me to, hey, if you really want to come to the business side of pharma, when you come out of your PhD, you should consider getting an MBA. And actually, one of the people who I had the opportunity to speak to about this was Mark Fishman, who at the time was the president of the Novartis Institutes for Biomedical Research. And Mark was invited by my program to chair one of our annual lectureship speaker series. I don't remember which one. We had like a million of them, but there was one we had every year. And that year we had Mark. And after the lecture, the graduate students in the department got to go to lunch with Mark. And obviously we like free food. So we went and we had lunch with Mark. And that's yet another thing that I would recommend to students. When you're in a graduate program, there are going to be a lot of notable scientists, entrepreneurs, business people who are going to be invited to your program to give a seminar. Even if you don't want to go to the seminar, you should go for the food because you don't know what you might learn in those sort of like post-seminar chats. So in chatting with Mark, I said to him like, hey, I'm in my third year of my PhD now, remember me from when I interned at NIBR 2 years ago? He was like, yeah, I remember you. You were in the intern program. You were in 2 consecutive intern classes. I was like, yeah. He was like, so what are you thinking of doing now? Do you want to come back to NIBR and do the postdoc program? Because Novartis is one of the companies that has actually a very solid postdoctoral fellowship program, which I had been considering. But I said to him, that I was interested in considering business development. And he said, okay, if you want to come into Novartis and business development, there are 2 paths you should think about. The one path is when you're done with your PhD, go down to Harvard Business School and get an MBA. Or when you're done with your PhD, go into management consulting, go and work at McKinsey for 2 years. That's literally what he said to me. And at the time, I didn't know what McKinsey was. I was like, what? What is this company where 2 years at this company is equivalent to an MBA degree? And so I started thinking like, okay, I definitely don't want more school when I'm done with this. So I automatically started considering the consultant path. I was fortunate to start a consultant club in my 4th year for PhD students who wanted to venture into management consulting or venture capital, invest in startups, or even entrepreneurship, starting companies. And so my brain started to kind of like shift a little bit. I was still scientist, but I was starting to think about new ways to sort of use my training. Along the way, I had the opportunity to work with a startup company simultaneous with grad school. So yet another thing I would recommend, again, only do it if your research is going well. When your research is going well, you can do a number of things outside the lab. When your research is not going well and it feels like
stay focused.
You kind of have to put away some of this stuff on the side. And I didn't get involved in any of these things until I felt like the research itself was truly an autopilot. Like I'll come in the morning, I just had to click a button, and then come at night and like wean the mice and like genotype the mice. Like, you know, it just was just doing itself. And so a number of internships in some startups in the Boston area started to like show me what it was like to work at a startup. And ultimately, coming out of my grad school, I graduated in March of 2016. And I took an offer from one of the top 3 management consultant companies in the world, Bain and Company. They had started hiring PhD students, PhD graduates alongside MBAs to help them, you know, deliver business solutions to a lot of your clients, many of whom are in the healthcare industry. So I was at Bain from 2016 to late 2017. And I left Bain at the end of 2017 to go found my own company. And so here I am today.
That's wonderful. And if you don't mind, because I've had a very similar experience, and I know it's a very different type of experience than being in academia. Walk us through how you conceived of what is your company's called 1910 Genetics?
Yes, 1910 Genetics. Yes, absolutely.
Yeah, how did you conceive of the idea and then getting out there raising money for it?
Right. So 1910 Genetics, we are a Cambridge, Massachusetts based biotech company. We're focused on leveraging artificial intelligence, machine learning, and a variety of other computational methodologies to accelerate the design phase of drug discovery. What I call the design phase, some of my pharmaceutical colleagues might call novel hit discovery, hits and lead optimization, et cetera. But basically early preclinical drug discovery. And so that's what we do. We're a platform company, we're a platform technology company. We're founded, officially incorporated in 2018, but I had been working on the company towards the end of 2017 when I left Bain and Company. And today, well, at the time of this recording, we're around a little under 15 people. And the people at the company are pretty diverse. PhDs in machine learning and physics, biology, chemistry. We got all kinds of people. And we have pharmaceutical veterans who have been at places like GSK for like 30 something years. On our team is medicinal chemists. So it's a truly interdisciplinary team. And we do the full phase, the full cycle of design, make, test, right? You design a drug using your AI algorithm or machine learning approach or whatever. You manufacture them. We do a lot of our manufacturing in China and India, in Ukraine, and we ship our drug candidates to our labs here in Cambridge, Massachusetts, and we test them, right? Because ultimately you say, does this drug candidate do what I intended it to do? So how did I conceive of the idea? Well, if you followed my story, my whole life, I've always known that I would be in pharmaceuticals.
Right.
Along the way, I picked up a business orientation, right? So it's not too hard to see how Jen is a pharmacologist by training, and she was a business person at Bain and Company, and now she started a company. It's not too hard to make that leap. What might be hard for you to envision is how, as a biologist, I founded a software company, because 1910 Genetics is foremost a software company. And it was born out of I've always been fascinated with technology, right? I've always been fascinated with some of my favorite companies in the world are actually tech companies out in Silicon Valley. I've always been fascinated by the pace of innovation and just the, how daring entrepreneurs in Silicon Valley are. They're very daring, I think daring is the word. And I had been studying and just following this rising wave of artificial intelligence, machine learning, this idea that you can leverage huge swaths of data that you have accumulated over many years and decades of doing experiments, and that you can learn from that data. You can train machines to learn from your past successes and failures and use that learning to inform your future design path for whatever drug, for whatever disease. The idea that all of your historically accumulated knowledge, both within your own organization and externally, can be leveraged to design better. We call it quality by intelligence, right? Aiming for the highest quality molecules in the first iteration of design, as opposed to the fifth iteration in the very first time you set out to design a molecule, that it's a quality molecule in the first try. And that's not what the case is using traditional approaches, where it's like, a huge team of medicinal chemists, they start, they design, and then they look at it, they design again, and then, we do some iteration, but our goal is to try to incorporate the broadest slate of quality parameters into a drug. So ensuring that it's potent, ensuring that it's selective for its target, ensuring that it has all the drug-like properties that you need, ensuring that it gets to the respective sites within the body where you want it to get to, and thinking of these things holistically in the first round of design. And with machines, you can do that. Machines can do two things at once. They can walk and chew gum, right? But a medicinal chemist is like, okay, no, right now, I just want to solve this potency problem. Let me try to make it potent, and then I'll solve the next problem. And it becomes this sequential discovery paradigm. You do this, and then you do this. But we're saying with machines, with the benefit of historical learnings, you can actually do many of those things at the same time.
So if you don't mind, talk to us about how you took your idea for the company, for 1910 Genetics, to an investor, because you had to raise money, right?
Yeah.
What was that like? I'm sure.
Right. What was that like? Well, consider the fact that somebody said this to me, I never actually looked into it. There are currently less than 50 Black women who have raised more than $1 million for startups in the US. I don't know how true that is. We've been in stealth mode. We haven't actually publicly launched. We've raised well over $1 million.
Congratulations, by the way.
Thank you. And what was it like? Look, I'm a praying person. I'm a Christian, so I'm just going to give God all the glory, is what I'm going to tell you. It was
but you can give God the glory, but somebody's got to write the check. So.
That's right. That's right. I do give God the glory. I'm going to tell you that. But it was hard. So practically, first of all, put your idea on paper, okay? Put your idea on paper. The business plan for what became 1910 Genetics, I wrote on long flights when I was a management consultant at Bain. I would have to travel to this client site. I would wait at the airport for 6 hours, 7 hours, because flights always get canceled. And I would just sit there and I would just open my laptop and I would just start typing. I said to myself, what does a pharmaceutical company look like when it's built from the ground up with machines, with the intersection of human and machines? What does that company look like? And I was just fascinated, and I was reading a lot about artificial intelligence as a discipline and how that whole discipline was evolving outside of pharmaceuticals in areas like driverless cars and autonomous vehicles and image recognition and search engines and just different areas. And I was thinking, some of these tools can be applied to chemistry. Some of these tools can be applied to drug discovery. And I would just keep typing, and sometimes I'll fall asleep, on the plane, and I'll wake up and I'll just keep doing that. So as an entrepreneur, you're a first-time entrepreneur, you're a young entrepreneur, put your ideas down, take the time to write a comprehensive business plan. If you're wondering what that's like, if you just literally Google how to write a business plan, you need some good starting points. If you are fortunate to be attending some of the schools in the area, Tufts has an entrepreneurship institute, the Gordon Institute of Entrepreneurship. I never went there, so I can actually speak to it, but I know it's a resource. Harvard has a big entrepreneurship center, so does MIT. But I also know there are a lot of students who don't attend Harvard, Tufts, and MIT, and who might not have some of these resources. So there's a lot of resources out there online as to how to put together a business plan. So the minute you write a business plan is basically your vision for the future in prose form. You write it in prose form. Once you write it in prose form, and I like prose form as the first way to write. I don't like to begin writing in PowerPoint, because PowerPoint is a sanitized view of the world. So it's important that you write first in prose form and you get a chance to express your thoughts. Then you make the PowerPoint deck, right? And then you think about how much money you want to raise. There are different rounds of financing. There's pre-seed, there's pre-seed, there's fruit, there's mustard, all kinds of names. I'm just kidding. It's roughly seed, Series A, Series B, Series C, et cetera. So when you started, you probably want to raise seed financing, right? Anywhere between $1 and $5 million. Now, How do you raise that money? If you got a good business plan, you got a good pitch deck that you've put together some advisors, right? People that have done this before, right? It could be people in grad school, and that's why internships are great. If you've interned at some pharmaceutical companies, ask some of those mentors whether they could be willing to serve as scientific advisors for you. Many of them can introduce you to investors, introduce you to resources, et cetera. And then finally, consider accelerator programs. We went through a Silicon Valley-based accelerator known as Y Combinator. Some of you might have heard of YC.
So Michael Seibel.
Yeah, Michael is actually, me and Michael just spoke last week about my current fundraising now for Series A. So he just actually introduced me to a very large investor because we're in the next phase. So I know Michael Seibel really well.
He's a good guy.
Sorry.
He's a good guy. He's been trying to help, particularly African American, or shall we say, the underestimated.
Michael is great, and YC has really doubled down in the life sciences. Yeah. So if you were thinking of YC as like, oh, they only fund tech, companies like Stripe and Airbnb, that's true. They did have those runaway successes like Airbnb and Stripe and Dropbox, et cetera. But over the past 3 to 4 years, they've been funding a lot of bio companies, and so they funded me. As a Black woman solo founder. Now, when you go out there, they'll tell you like, oh, they don't like solo founder companies. That is true. It just means your work is, you have twice the amount of work to do. So if you do have a great co-founder, great. But what my advice to entrepreneurs is, don't bring on a co-founder just cause, right? Because you're actually better off not having a co-founder than having like a co-founder who is not a fit. And then now you got to fire that person, or you guys got to break up. And like, founder breakups are the #1 reason for Y Combinator companies failing. Yeah. Literally. So find a partner if you've known them before. YC has found that the founders who tend to make it are those who have worked with one another in a prior setting, right, prior to starting the company. So sometimes people are like on some of these social networks, like, oh, I found somebody, I think we want to do a startup together. It's like, do you know this person? No. You met them before? No. Well, this isn't Tinder, right? Find somebody that you've worked with, that you've interned with. Perhaps you went to grad school together, or you went to undergrad together, someone you respect, right? There's got to be someone where you're thinking like, you know what? I think this guy is the smartest engineer that I know, right? Or this person is like the smartest biology person that I know. So I'd recommend that you come at it from the perspective of, yes, it'll be great if there's someone who's willing to commit to realizing your vision. If you can find that person, bring them on. If you cannot, like in my case, I could not, whatever the case was, I went to Y Combinator alone. They funded the startup. We went through the accelerator program. We came out, we closed a sizable seed round. We came back to Boston, grew the firm. Now we're raising the Series A financing, and we're getting ready for our public launch, press launch.
And that was my next question. So when do you project that 1910 Genetics would have its first launch, public launch?
In about, if it wasn't for the election tomorrow, we're supposed to launch last Tuesday. We delayed it because we didn't want to get mixed up with the election cycle news, but I think we'll be launching, I don't know, first week of December or so.
Gotcha.
Yeah, a lot of companies, when they come out of Y Combinator, they launch immediately. We get a chance to announce the seed round and all of that. I didn't think we needed the press at that time, because the focus for me was just to double down and build the technology, because it's kind of like, what do you need the media for? So focused on hiring. So I went to Silicon Valley for 3 months, raised the seed round, and came back. And I focused on hiring. Like I said, the team is very engineer heavy, biology, chemistry, hiring across those first 10 hires across a variety of disciplines, and just hunker down and build the first version of your product, what we like to call the Roadster, kind of like when Tesla started, like the first version of the car, the Roadster. Build the Roadster of what it is that you say you're going to actually build, and then you can open it up to the world. So that's the stage we're at right now.
How did you come up with the name 1910 Genetics? What's the symbol?
That's a very common question. So 1910 is the year that the first patient with sickle cell anemia in the US was diagnosed.
Oh, I didn't know that.
Yeah, as a young guy, West African, actually from the West Indies, from Grenada, from the West Indies. And obviously sickle cell disease takes its root in Africa. And the genetics in our name is actually short for human genetics. And that means that we try to work on diseases for which there's a strong human genetics validation for the basis of the disease. So if there's a human genetics component to the disease, we like diseases like that. But also, even if there isn't a human genetics component, if there is a clinical validation of the therapeutic target of interest, we feel more confident in bringing our platform to bear. One of my mentors in pharma said, Jen, the best quality molecule cannot save you if the biological target is wrong, right?
Yeah.
So it's our way of saying we are fundamentally a technology platform company focused on doing chemistry and creating quality molecules via intelligence. But those molecules are only as good as the therapeutic target, the disease target, the biological target of interest. Right. And so having some North Star as to how we select therapeutic targets, using human genetics as that North Star. And sickle cell anemia is an example of a disease with a molecular basis, right? The first time that we knew that a disease could have a genetic basis was sickle cell. Right. So we're not a sickle cell company or anything like that. It's just, the name is just a play on words and just trying to pay homage to our North Star and how we select diseases to work on, how we select targets, therapeutic targets within those diseases to work on, and then how we're bringing our platform to make undruggable targets a thing of the past.
That's absolutely wonderful. Well, I want to thank you, Dr. Nwankwo, that for you to spend some time with us. I know you're very busy. Your passion for biology, your passion for research, but your passion for making drugs more efficiently and effectively is just powerful. Thank you for sharing your HBCU roots and how that's connected to everything that you do. We wish you the best. We're looking forward to seeing you ring the bell on Wall Street at one point. So don't forget about us little people when you become big and famous. So
no, you guys are not little. Thank you so much. Really appreciate you taking the time. Thank you to UNCF. Thank you. You asked how Claflin prepared me. I did get the Medtronic Foundation scholarship from UNCF when I was a junior. So that was helpful. I was able to buy a couple of laptops, some books, and just helped me sort of keep my head down and focus on my academics. So thank you to the United Negro College Fund. You guys are doing really important work. And thank you.
No, appreciate it. Appreciate you. I'm looking forward to following up with you in the future too. So thank you.
Likewise. Thank you so much, Chad. Thank you all.
You're welcome. That was Dr. Jen Nwankwo, who is the president, CEO, and founder of 1910 Genetics.